Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Hello, everyone. I'm excited to be here today with Martin Borch Jensen, who's the co-founder and chief scientific officer at Gordian Biotechnology, and he's also president of the nonprofit Norn Group, which focuses on therapeutics R&D for anti-aging. We're going to talk about the state of the field of aging and anti-aging, and also how this is relevant to drug development, not just for the kind of broad goal of having a healthier and longer lifespan, but why this is relevant for almost every area of therapeutic development. So Martin, I'm really looking forward to it, and thanks so much for joining us.
Yeah, thanks for having me.
I can't help but ask to start with a really interesting story that is, I think, related in some way to you getting into this field, which begins with you eating so much at Indian buffet that you couldn't eat for an entire day, and ends with you writing a book about intermittent fasting. So maybe you could talk through that story and how that connects to what you're doing.
Yeah, sure. So I was already in the field. This is while I was doing my PhD at the National Institute on Aging in Baltimore. So I had already decided that sort of aging biology was the thing I wanted to focus on as sort of potentially the biggest lever we have on. So anyway, I'm doing the PhD. I'm focused not on sort of intermittent fasting, caloric restriction as a topic, but on DNA damage and ways that this can drive the aging process. But I'm reading the literature, right? I'm aware of all this stuff. And then, at some point, I guess there were like two things that converged. One was like, okay, so I'm reading about this stuff and it works in mice, and like there's all this, like what am I doing if I'm an expert in the biology of aging and the different things that like work in animal models, and I'm not even thinking about,
Right.
like is
Not
this
doing it
ready
myself.
for humans,
Yeah.
right? So that's sort of in the background, right? And then the triggering point, as you said, was some Indian buffet on a Saturday with the other postdocs in the lab in Baltimore. And, I like Indian food a lot. So yeah, I ate enough that I got up and I just skipped breakfast. I wasn't really hungry. And then by 2 p.m., I hadn't really gotten hungry yet. And so I figured, well, maybe I should just try this thing, right? Maybe I should try this thing called alternate day fasting, which, in mice and rats, they basically, you give them food only every other day. You don't restrict what they eat on the days when they're eating, right? So the overall food intake is not necessarily restricted, but there are periods of fasting. And in the animals, it sort of improves metabolic health and makes them live longer. So I decided, okay, let's just try this. And it worked out. The next day I was really hungry, wake up a little earlier and have a larger breakfast. But I ended up doing it, for 2.5 years during my PhD. And then, I'm from Denmark. So I was doing my PhD in Baltimore. I got back to Denmark. I had to do sort of mandatory military service. We still have conscription. And so I was back, and then, some friend of a friend was a journalist at one of the Danish newspapers and had heard about this thing and was like, oh, that's a little weird. But not only is it weird, like the guy has a PhD in this stuff. So it's maybe like the right kind of weird. And so they ended up sort of like writing some newspaper article. And then I think, as sometimes happens, other people are like, oh, this is indeed the right kind of weird. Let's like write some more stuff. And then a radio show was like, oh, we want to interview you about this. And then eventually it culminated with a publisher was like, oh, this should be a book. This was right around the same time that Michael Mosby was writing this 5-2 diet book that was sort of also on the theme of fasting, although a bit more easier for people to do, or sort of a light version.
Yeah. Two-part follow-up question to that. One is, I think it's well established that intermittent fasting extends healthy lifespan in animals, in many animals. Is it also the case in humans? If not, or if it hasn't been established, why don't we know, or what do we know? And then related to that, what's the mechanism behind it? Like why I think we have good reason to believe it works in mice. What's behind that?
Yeah, so there isn't like a single, here's the one thing that's happening in the animals. There's a whole bunch of things that may be beneficial that are, so obviously, like if you have insulin resistance, that's bad and you'll have diabetes and so forth, but you also have like, if you have fat accumulation, there's higher chronic inflammation. So there's sort of a range of things that are generally correlated with healthier states that are changed. Nobody has like this is the mechanism for caloric restriction, and it's not obvious that there would be like a single mechanism as
Yeah.
opposed to, personally I think a lot of the things we have in the aging field that have an effect on lifespan, whether it's in mice or monkeys or whatever, they relate to nutrient sensing, more often than not, 3 times out of 4. And personally I think that that's because you find a master switch. So like aging is a big systems emergent biology thing. It's not just like a single process with
Right.
like 17 genes involved. And so, in order to really move the needle, I think finding sort of like evolutionarily things present in our genome that are like suited for certain states, and then you find those knobs and then you can turn them. That's, like I can't prove that, but that's sort of my take
Yeah.
on. But then that is also the reason why there's like a bunch of mechanisms involved. Okay, so does it work in humans? What is caloric restriction, right? It's like often defined as you give the animals ad lib like as much as they want to eat, and then you restrict, so whatever they end up eating in that regime, you restrict that 20 or 30% or some other number, but
Yeah.
typically there. So what does that mean to a human? Like, are you eating ad lib right now? Are you already like 20% restricted because like social pressures around, like weight gain, all those kinds
Yeah.
of things? So like, what is the right dose of caloric restriction in a human or fasting, right? I think that's what we don't know. And this really mirrors basically drug development, which I think we'll talk about later, right? The hard part about the human trial is there's so many things that you don't know how exactly should we design this, right? Like it's not just picking a drug and then saying, like this is the only variable, but it's like, what is your dosing? What's your duration? Like who are the exact patients and so forth. So for caloric restriction, there's studies in different organisms. I think it's been done in mice, flies, and yeast at least, where this sort of restriction regime is applied to genetically diverse, either wild animal population or outbred animals. And there, the same dose will sometimes produce good effect, sometimes produce a bad effect. And so I think finding the right dose seems tricky in humans. I would guess that, like being hungry a reasonable amount of the time is probably like
Yeah.
the right dose is somewhere around there. If you're never hungry, yes, caloric restriction will probably work in you. But
Right.
optimizing it beyond that for people who are sort of exercise a bunch and so forth, the right regime is seems tricky. And this of course comes, back to for the aging field in general, how do we measure whether something is working? I think that's really the number one barrier for the aging field is like, what is the biomarker that tells you whether you're doing better or not? Otherwise, it's hard to optimize.
Yeah. Yeah, that's a great segue in where I wanted to get to, because obviously caloric restriction, intermittent fasting is a small sliver of the potential interventions that we could think about to have healthier aging. But it begs the question of how do you even measure it, and what do we understand about it in the first place? Because I think for people like me who aren't deeply in this field, you have the very high level question of, is healthy aging just lack of the diseases that we can think of that are all aging associated, like cancer and Alzheimer's and so on? Or is there a more fundamental unit? We had Austin Argentieri from Harvard and Broad Institute talking about using a proteomic signature for measuring biological age. There's been a lot of other kinds of signatures of measuring biological age as opposed to chronological age. Maybe you could talk a little bit about what we understand and what we don't understand about just the fundamentals of aging. And then I'd love to just hear about how that influences everything else in drug development, because it seems like it's a core thread that runs through the middle.
Yeah, yeah, that makes a lot of sense. So what do we know? We know that it's a biological process that is malleable, right? Like you can change the rate of aging. We also know that it's like the number one risk factor for 7 or 8 out of the, top causes of mortality in the US and more broadly, and the number two for sort of the rest. So again, like I said before, like definitely biggest lever on health. If everyone was like biologically 28, we wouldn't need a whole, like we need like a little department for like testicular cancer research and like a
Yeah.
couple of other cancers that happen when you're younger in both sexes. But mostly cancer would just be irrelevant or very rare,
Yeah.
like incidents. And so, okay, can we get there? So then we know, well, it definitely can be changed. I mean, I think there's sort of like two ways of knowing that. One is we've done it a bunch of in animals, right? Like you can make animals have different trajectories. You can do that even with interventions that are started relatively late in life. And then also, philosophically, like you can have a kid. Like one cell from your body can create a completely young human body, with the
Mm.
help of one cell from somebody else's body. And so it's like we have the sequence of events that creates a young organ body, whatever, like it's definitely possible from within, your genome and the basic components of a human cell. So that's just like, is there anything there? So then the question is, where are we now? And really if you go back 50 years, the aging field was just like, do cells age or is that not a thing? Like, are the
Right.
cells all the same? Like we didn't have the tools, right? Then
Yeah.
around 1990, we find the first genes that can radically extend lifespan in C. elegans worms. And so, okay, we can do a thing here. Like you can change the trajectory quite a lot. And then sort of in the 2000s, 2010s, there's different yields or different subsections of the aging field that sort of get steam. There's this whole, there's stuff in blood that seems to make us younger or older. And there's like whole research angles on like mitochondrial function and these proteins and that proteins and so forth. And so that's really laying out what currently would be like, what are the topics at a conference? Those are all things that happened usually in like the last 20 years, sometimes less. So what is the unifying theory of how aging works? It doesn't exist. Or, I mean, a few exist, but none are like
None are
clearly.
rock solid. Yeah.
Yeah, exactly. Like, what is the thing that is actually happening? We're not there yet. And part of that, I think, is because there are a lot of things happening and the things are connected, right? So it's not like, it does not seem to be the case that aging is a like linear sum of individual sort of like linear processes, right? Like
Yeah.
things are connected. If you have some, inflammatory state that leads to like a bunch of cell death in one organ, that organ, of course, is sending stuff out to the rest of the body. And so now other organs are behaving differently and so forth. So I think it's more of an emergent property of a system. Personally, I would say we should think about the extent to which the system is appropriately calibrated internally rather than just like what are the individual nodes of the things in the system, right? Because in biology in general, like more is never like good or bad except in
Mm.
a specific context. So, right, it
Yeah.
depends. Like, is inflammation good or bad? Well, it depends. Did you, get a worm into your, through your food on a vacation or whatever?
Yeah.
Um, so, so I think, yeah, we don't have the like, this is the thing. What we do have are a handful of different ways that we can extend lifespan in animal models through different arms of biology. So not just one thing, like there is this nutrient sensing heavy axis,
Yeah.
but there's also this sort of like dysfunctional cell, maybe senescent cells, which is not like a super clearly defined, like what is a senescent cell? It's probably a spectrum of things, but sort of an inflammation axis there that also overlaps with adiposity. And then there's like specific dysfunctions in terms of like DNA damage things, they tie to those and so forth. So there's a bunch of stuff that you can do. We're finding ways of like, let's do this thing and then see if like that's improving. And the gold standard for like an aging paper is one that has like a Gompertz curve, like lifespan study and like
Yeah.
shows some shift there for the most part, right? Because and that's because we have these ways of like measuring biological age, but none of them have been validated to be predictive of perturbations, right? So that's an important thing. You can find something that like correlates with, okay, the people who died sooner, they had this phenotype
Right.
versus people who died later. So we have these different methylation clocks and protein clocks and so forth that predict those things. And that's like a good first step. But that doesn't necessarily mean that if you make A look like B, B will now live as long as A, right?
Right.
So that test is
Yeah. Is it a cause or a consequence of something else? Right. That
right.
clock. Yeah. Yeah. It's kind of like if you change the hand of the clock to say a different time, it doesn't change the time, right? You've just
Yeah.
changed the hand of your clock.
Yeah,
Yeah.
exactly. And most of the clocks are, well, many of them are sort of like black box, right? Like you have some methylation sites and they're just like picked by machine learning algorithm from like a bunch of changes, right? And so you don't know the meaning of any individual thing. This is a general problem in biology, right? And I'm sure your listeners will
Yeah.
appreciate, like you have genetic data or transcriptomic data or whatever. Okay, things are changing. And like, what does that mean? Where does it fit into the system? And the best way to know that, maybe, well, I don't know, the best and the worst way to know that is to understand everything about how the system works. Then you could just say, oh, this changed. So then this is what's going to happen. And of course,
Yeah.
we don't know that. And we want to do things before we know that, which is also, like the company Gordian Biotechnology, why is it called Gordian? The history of the
Not.
Gordian knot, which is like impossibly complex knot that, people want to untangle because the Oracle of Delphi said that whoever unties this knot will go on to take over the world. And you can't do it, right? So that's me in academia, for 10 years, trying to figure out how does aging work? Like, if I just understand the whole thing, then I'll know what to do. And then eventually it's like, no, this is not the way. Right. And so, in homage to sort of Alexander, the not yet great, who cut the knot with his sword in most myths, and then, yeah, made a bid for taking over the world, right? Like you solve it a different way, which for us is this in vivo pool screening. But like in general, if you have a full, like complex, multidimensional biological system, how do you find out what is the effect of a given perturbation without having to understand the whole system? Right.
Yeah.
And you want to know that because for not everyone, some people are just curious, but like we have A and we want to create B, right? Like you have a sick person, you want to create a healthy person. And fundamentally, you want like some sort of magic 8-ball machine that tells you, okay, I want to go from A to B. What do I need to do? Or at least that's, what we want at Gordian. But I think
Yeah.
that's what any drug development company wants, right?
Yeah. Yeah. So talk about this in vivo screening approach. I love the name and I love the kind of idea that it evokes. So you have this completely entangled knot, and the only way to undo it is to cut through it. So yeah, tell me about this approach and how you propose to cut through it.
Yeah, so I think, I'll talk about both like the why and the how, right? And so maybe we start with the how. To people who sort of like are doing drug discovery or any kind of like discovery biology, that like a lot of the time you want to see, can we bring A to B, and you run an experiment, right? Like you give this thing to a bunch of mice and you see if they get healthier. So the A and B that we wanted to solve are, conditions of aging, right? Like all the things that go wrong with aging. So we work in 4 different disease areas: heart failure, lung and liver fibrosis, and osteoarthritis. So for any given one of these, you have a cell inside your liver that is not happy, right? Like you've got steatohepatitis or whatever, and you want to see, how do we get over here?
Yeah.
And so if you know what to do, it's easy. If you don't know what to do, then the default approach is some sort of like high throughput screening, or, trying to understand it better and then do a targeted screening, right? And so you try a lot of things. That's what we wanted to do as well. But the diseases that we want to fix, they involve biology of like different, it's not just your hepatocyte, like that's not in a good state. It's interacting with the Kupffer cells and infiltrating immune cells in the liver, right? And like there's all this stuff going on that's coming in through the immune system, through other like interactions with resident cells, interactions with the extracellular matrix. You get fibrosis and it gets stiffening, interactions with your sort of metabolic state. The accumulation of like excess fat in the liver is usually the trigger for this non-alcoholic steatohepatitis. And so you kind of want, like, if you don't know exactly what to do, you could ask in an isolated hepatocyte, like what makes it more like B? But is that relevant if you're not starting in like this isolated hepatocyte in a plastic dish state, but you're starting with like this complicated in vivo state, right? And especially for these age-related diseases, we found a lot of ways to cure Alzheimer's, quote unquote, in
The
young mice
dish.
that
Yes.
are overexpressing like 5 specific mutations of, a beta. And so you can cure that thing. And is that your patients or not, right? And so I think the diseases that we're going after, you could get to that list of indications in sort of at least 3 different ways. One is you say, oh, these are complex diseases of aging where in vivo is very important. That's one. You could also say, never mind aging, they're just the biggest unmet, like medical needs. And then you could also say, like, yeah, they're the ones where the models for drug discovery are least like the actual patients, right? Like what we can normally do is least like that. And so they all point you in the same direction, not coincidentally, right? Like they're the unmet needs because we haven't treated them, and
Right.
then plausibly because we haven't simulated them well or emulated them well, right? So that's sort of what we're going after. And you can, like we have these big unmet medical needs where generally like trials fail a lot. We need to do something different. And so this is Gordian's version of doing something different is let's create these avatars. Like let's get really close to the context that we want our treatments to
Yeah.
work in. And then see what works in that context with whatever levers are broken and like sand and gears and stuff happens with this aging process. And then yeah, what works there. And so the way that's like, what are we doing and why are we doing that? And why is that relevant? I
Yeah.
think more broadly to, drug discovery, right? Like anyone else who's studying a disease that only happens in old people. Well, what is the biology that makes it only happen in old people? And is that part of your sort of like test system? And then the way that we do it is we turn the diseased organ with all the like full complexity of like in vivo disease into sort of a series of cellular perturbation experiments. And so this is sort of similar conceptually to having like a 384 well plate. And so you've got 384 divided by number of replicates, different experiments.
Yeah.
And they're independent because it's a different batch of media separated by like a plastic well, right? And so that's good because you can actually tell what's going on, which of these perturbations was good. And of course, it's bad because the plastic well is not normally what's going on, right?
Yeah.
And so what we do instead is use AAV gene therapy to deliver perturbations at a very much subsaturating rate so that most of the tissue is unperturbed. And then these islands of perturbation exist throughout the tissue where a cell is in its full disease context and it's, got this gene A upregulated or downregulated as a
Yeah.
potential perturbation, therapeutic perturbation, right? And so, yeah, you have these like, and then it's divided. It's independent from this other perturbation because there's a whole bunch of other diseased cell tissue just like in between them, right? And this sort of like radius of effect of any given cell is not as big. So you have to calibrate that, and that's a bunch of the sort of hard parts of making this work. So that creates these perturbations. Then the question is like, how do we read it out? And there's two parts to that. One is like, how do you get data? And then one is how do you interpret data? How do you get data? So this is basically we're sort of like riding on the wave of single cell transcriptomic, like high throughput assays. So we use, give a lot of money to 10x Genomics, for example, right? And like they're sort of
Yeah.
every time they do better and make a new assay or something, our stuff becomes cheaper. So that's very much by design and a happy sort of like state of coexistence. And so what that gives us is, you can say a phenotypic readout. So if you screen, right, you've either got like, okay, we've already decided what the target is and you're looking for target engagement, or you've decided what the state you want to end up in is the phenotype, and then you like screen for that phenotype. And so we use the whole transcriptome as like a phenotypic readout. But sometimes I call it a phenotarget readout because we're using like a genetic therapy, we actually know exactly what happened. Unlike in one of the downsides of phenotypic screening is you get all these compounds, you put them in, whatever, your tanks of zebrafish, and then you see if any of them like flip out or whatever, turn purple. And then, okay, cool, this did. How did that happen? We have no idea, right? So our advantage here is that like there's a more tightly integrated loop between the transcriptome and then the like genetic perturbations we're doing.
Yeah. And
But
all of this, just to clarify, all of this is happening like it's in like a non-human primate or a mouse or where is all this happening?
Yeah, so it's happening inside a whatever the best animal model we can find for this disease is. So we have experiments in rodents. We also have experiments in large animals. And then it's not just like what's the species. So species matters. I think there's a whole like, the mice are to blame for every failure in drug discovery. I think that's wrong.
Or the overinterpreted phase 2 trials. It's another candidate.
Yeah, and then, but it's like, okay, but how do you use the mice? So if you're studying osteoarthritis and you're going after, 75-year-old patients who have developed osteoarthritis over decades, and your mouse model is like 8 weeks old and it's males only, and you've like destabilized their joints in order to rapidly induce, cartilage damage.
Right.
It's not the same thing. Maybe it's the mouse's fault because, like there is less cartilage. There are definitely physiological differences, but maybe it's like you didn't model half the factors in disease development, right?
Yeah.
So are lazy. So anyway, if you use a racehorse that has developed osteoarthritis by running around over an extended period of time, you alleviate both of those. You both have
Yeah.
like more human-like cartilage thickness and composition and weight on the joint. You also have sort of a more human-like etiology of disease. And so for
Yeah.
each of the indications, there's different models that are like more appropriate. And we try to do our screening in whatever is most like the patients.
Yeah. Yeah, this part is really interesting to me because one of the things that caught my eye about what you do besides the approach, which I think is really interesting, is also from a humanity perspective. I think we want to use as few animals as we can to get like I think it's unrealistic to remove animals from the drug development process. Obviously that would introduce downstream dramatic negative consequences for the people that things are tested on. So given that we have to use animal models and primate models and those sorts of things, we want to make the most that we absolutely can. And what I didn't appreciate is that you can also potentially do these kind of things in aging race horses or some other kinds of models that mean you don't have to necessarily breed animals specifically for this purpose. So I was curious about whether there's an end game to this where you may be able to think about how you model what you do see in a non-human primate, for example, and you can start to do these kind of experiments in silico, because the more data that you collect, the more you might be able to say, look, we've done this process in hundreds of monkey livers to study MASH or other liver diseases, and so actually now we have a pretty good understanding of, we can predict a priori without having to expose a monkey what might be happening. So I'm wondering if that's kind of part of this process in the long term as well. We can build just better models of preclinical understanding.
Yeah, that's definitely a plan. We definitely do not have like 100 monkey livers.
Yeah.
But everything you said makes perfect sense, right? And I agree, at least for the diseases we're in, you can't, if you eliminated animal testing, you have to accept a lot of like unexpected stuff happening in human trials. Is that trade-off you want to, I mean, that's up to, like the FDA and ethicists and whatever, right? But there's no doubt that you cannot model the different interacting pieces like the immune system in a system where like some sort
Yeah.
of like organ on a chip where that's just like absent, right? And the biology is too complex since we don't know how it works, we cannot create the perfect model that includes all the relevant stuff, right? But in a reductionist simplified way. So I think that's right. For sure, they're like in silico. I mean, we're basically like right now we are generating a data set that, definitely doesn't exist in the world. I was just at a like AI foundation yesterday talking about this stuff, right? And they're like, oh, can we train on this stuff? And I'm like, well, you can train on CMAP or whatever, like a bunch of cancer cells in a dish with chemicals on them. You cannot train on like what is the logic of like in vivo biology with perturbations yet. I mean, we're starting to be able to do that. So right now we're definitely generating a lot of data for what will happen, and then we're using that to like, that's a recursive thing where we use that to make better predictions that tells us what to screen and tells us what could be combinations and so forth. One day entirely in silico, I'm sure, but like, is that 230 years from now? Maybe. Depends on how complex biology is, which I don't know yet. All I know is like, I think we know, there's more we don't know than like we know in
Yeah.
terms of how does like everything fit together. So the more progress, the better there. But Gordian is set up in a way where it's like, if that takes a long time, we're good. We're just testing the perturbations. We're finding the therapies, right? And if
Yeah.
we become able to do that sooner rather than later, then we're also good, right? And we'll probably get to do that, even sooner. There's also an interim step there that you could think about, which is just kind of going on a mild tangent here. But if you can keep so let's say like somebody dies and they're organ donor and they donate their tissue and they have a fibrotic lung. And so it cannot be used for transplantation. And so they've donated their tissue for research. So often, you can like, and we have agreements with donor organizations there where we'll like go in and like molecularly characterize the lung and try to figure out, like how is this similar or different to different animal models? That's part of how we pick, like, what are the most relevant models? We had some interesting stuff there in lung fibrosis, actually, where some of the models that like histologically look like human are not actually keep that lung alive for a month or something post-mortem. And then you could do your pulled in vivo screen inside like a living organ, a human organ, right? So that's anyway the closest thing
Yeah.
you can get to. There's some, there's some caveats to it still, but I think that might be like an intermediate step of where you like either advance from or complement the sort of animals with like actual human. The main problem there, I think, is like keeping the organs alive ex
Yeah.
vivo for an appropriate.
Yeah, that's fascinating. Kind of coming back to the topic of aging, question that maybe sounds a little ridiculous, but I hopefully I think there's something there. We've been seeing more and more of tech people like Brian Johnson and those sorts of, and I think there's now one of these things where there's a chronological age scoreboard that you can get one of these, chronological age tests regularly and submit what your biological age is according to the test and the kinds of things you're doing to bring that score down. And I struggle a little bit with like whether there is something there. And actually, these people who have seemingly all the resources in the world to go and understand the science and put together a cocktail for themselves to really understand like what can I do to extend my age? How much of that is solid science? How much of that is we actually don't know, and they're doing the experiment in some ways in public in real time. I'm just really curious to get your take as somebody who spends a lot of time in this field of like what can we make of that phenomenon that seems to be growing and growing.
Yeah. I mean, I think generally competition can be very good for forcing people to like find efficient strategies, right? Like I play a lot of board games and like strategy board games, right? And like, yeah, you put into an environment and then you're like trying to find how to create. So that part's good. In terms of like, is it solid or not? I think it's a mixture. There are people who are very serious about like finding what is the best documented research on some drug like rapamycin, let's say, which I think is probably currently the like prime candidate for like an aging drug that could be beneficial if used sort of at the right dose. And so we're back to like, what is the right dose? The main challenge for this thing is like the score metric hasn't undergone the kind of like quality assurance you would need, for example, for it to become like an FDA surrogate endpoint. Just back to what I was saying before. You have some, and so there's different scores and so forth, but like you have some methylation sites, they change that correlates with either for some of them, it just correlates with your chronological age. For some of them, it correlates with within this cohort or these two cohorts of patients, like time until death is sort of like correlated with this thing. But as we were talking about earlier, we don't know how those clocks respond to perturbation. And so what's the specificity of this readout, right? Like one assumes that you could find some compound that like changes your DNA methylase expression or something like that. And then you're going to get a bunch of changes in like clock sites, and it's going to tell you some age that's like, wow. So I think that's one thing that just like hasn't really been explored super well. And then there's also just like how well is the individual assay? Like, just if you were going to do this, right? Like I would recommend that and especially if you have like a lot of money, just take the same sample and like submit it 10 times or something. And then like what's the variability get in your ages, right? And so like that it is important to know if you're going to make decisions of like, oh, I started eating more of this stuff or that stuff, and like should I trust that my age is down? So I think, it would be great if making a leaderboard pool, and then if someone got together and said like, let's really do like a third party evaluation of putative aging clocks ability to predict sort of like rejuvenation, whatever you want to call it, like positive effects on biological age. And I think, this is kind of stuff we think about at Norn, and we're like probably actually on this very topic, which you didn't know, sort of putting out a call to action for like the clock field of like, okay, you could partner with so there's something called the Intervention Testing Program sponsored by the National Institute on Aging. And so this is like the gold standard for, will this thing make mice live longer? So you can submit a proposal for a compound, and then they'll test it. They'll test it at 3 different sites with matched protocols, but like independently and in males and females and like well-controlled, like control groups. And so that's another problem of like, are you fixing a sickly, mouse population, or are you actually extending from like best practices? So anyway, that exists. That's existed for a decade or something. And they have positive data here and there, and then they have negative data on various things that like people think might work. Okay, so like use blood data from like that cohort. The ITP is not aware that, like I'm suggesting this partnership with them, but
It's coming their
yeah,
way soon.
it might
Be
be, yeah,
prepared.
exactly, right? But like you could say, oh, if you have a new clock, let's have the like NIA clock benchmarking program, and you get some data from different mice in these cohorts, right?
Yeah.
And then your job is to correctly predict the perturbations, like which of these perturbations are positive and negative. And so they withhold a bunch of training data that nobody ever sees outside of the Rich Miller or whatever the groups, right? And then third party. And then the better your clock actually does at predicting mouse lifespan, the more we should like put that clock up as a leaderboard. And if it's like random dice at predicting that, then I don't know, maybe there's some mild shift towards healthier states when people are optimizing for this thing, but I don't know.
Yeah. How much is the lack of a business model around slowing aging a problem in the field? Because even in like I live in the UK normally, and with a cradle to grave national healthcare system, you could imagine in theory that the system might pay for healthy age. You could abstract away this. Are we treating cancer? Are we treating Alzheimer's? The point you made earlier, if we could dramatically slow down aging for everyone, we would solve a big chunk of the cancer problem. But the health system, even in the UK, where you have a kind of cradle to grave system, you can't get paid for that outcome right now. And you certainly couldn't in the US get reimbursed for slowing aging, probably because of this measurement, this surrogate endpoint issue that you raised. But does that slow the field down and force companies like you to say, actually, we can't solve the core aging problem. We need to focus on solving diseases that are the consequence of aging, or is going after the diseases that are the consequence of aging kind of the natural way to think about this anyways?
No, I think big picture, it would be dramatically better to have a metric that predicts a whole range of diseases and then test drugs against that, right? Like, so like the way clinical trials is, you have sort of infinity number of candidate drugs, and then you have like 10,000 diseases, give or take, right? And then you're sort of like doing these one-to-one
Right,
tests.
right.
So
Yeah.
that is a large combinatorial
Yeah.
problem. So I very strongly, solidly established them as predictive for different diseases. We should, like, if the UK government should force those to be part of all clinical trials. And the trial
Yeah.
runners, there's an expense. The expense is not huge, like extra blood draws and some omics or whatever on if you're already running the trial. In most cases, it's like less than 10% added trial cost. So it's not trivial, but like it's not that bad. And you could test against multiple diseases with like this data, right? I think the bigger reason you wouldn't want to do this as a pharma company is like, oh, if anything looks worse, now your drug might not be approved for the
Right.
primary. And so like I don't really fault them. It's
Mm,
on the one hand, it's like, oh, this is bad. We're doing like local optimization and like global not
right.
necessarily, right? But, you do have phase fours for assessing the safety, but I don't really fault it because it's like so hard. Like you can start a biotech company, you can go for 10 years or 20 years and like devote a third of your life to doing this thing. And you just barely like get an approval or not an approval, right? And so the consequence of tightening things is just like fewer drugs. And I think there's a thing there around like, okay, I think we're too accepting of status quo, which is like thousands and thousands of people dying versus like there are risks in trials. But anyway, so if we had a system where every time we test a drug, we test it for broadly for like health improving effects and like multiple diseases, you would search the space of like potential things that benefit humans dramatically faster. And so I think that that is a shift that should happen. Now,
Yeah.
even if that shift happens sort of like policy wise, and it definitely should happen in like, yeah, as you said, cradle to grave healthcare systems, because you're stuck with the bill anyway. In the US, there's sort of this like weird sort of like hand the problem to somebody else,
Yeah.
incentive. You still need the predictors, right? Like, so you need to be able to actually, and it's not like we have a test right now that you could just run and it gives you the like risk of every disease, and then And we know that the test is responsive. So I think, back to the sort of for humans as well, like we should invest effort, in my opinion, we should invest sort of like government nonprofit effort into creating predictors for multimorbidity, for like just overall health state. And then we should like absolutely jam those like into every clinical trial.
Yeah.
That's gonna 10x, like how many drugs do we find per year or whatever. But
Yeah.
that's complicated. So back to the question for the aging thing. I mean, one, you don't have that test. And so I think that's a bigger, like aging is an indication. It's not that like the NHS is like backwards and don't accept it. I think it's more like, okay, how are you going to measure the thing? Right?
Yeah.
Like what is the endpoint of your trial? And we don't have a amazing thing. And there's some things and, like intrinsic capacity is in ICD-11 for the WHO and stuff. So like there may be some movement towards there. But I think that would be, if we had that, that would be amazing. And if we had that and then people are like, no, you got to treat individual diseases, then that's real bad, right? Like you're just killing people for sure. So I think it's more like, I think the aging field is more hamstrung by technological advances than by the true and negative fact that we have developed sort of a siloed organs, individual organs sort of mindset for disease through, I don't even know the full history, right? But certainly the NIH in the US, there's like institute per organ or a couple of organs, right? And
Yeah.
like in
Yeah.
medical school, you'll become a cardiologist or whatever. And we don't have like an inflammationologist, right?
Right.
Could be an immunologist, but like, so I think that we're at a stage now where we have treated diseases that fit into certain specialties. We've done very well at like infectious disease. And that's probably because like the label was actually accurate for a
Right.
number of things that behave the same, right? And obviously, like, yes, a cardiologist knows a bunch about the heart, and that is great. And we should have non-zero number of cardiologists. But maybe at this point, the diseases that are really plaguing us span broader biology, and we should have experts in that and initiatives guided towards that, right? Like we
Yeah.
should have people focused on inflammation.
This has been fascinating. I could talk to you for hours, but we don't have hours today, but I'd love to do this again in the future. If people want to keep track of you, your work, you're at Gordian.bio, I believe is the website. But what kinds of people do you want to hear from? Are you looking for partners? Are you hiring? It'd be great to just hear if you want anybody to get in touch, who would that be?
Yeah, we're definitely hiring right now. So that we have a director of genomics role open, like people to focus on our single cell biology, wet lab, dry lab, interface. So that's a role. And we're also starting to talk to people for sort of translational genomics, thinking about the sort of like animal to human that we're already thinking about, but sort of like how do we know that this measurement in the animal, how do we implement that in a trial and stuff? So I think those are two roles that are pretty relevant for the audience, I think. And then, yeah, I mean, we're going to be a drug development company that puts things into clinical trials. And I think early on, we're definitely planning to do that in partnership with pharma companies. So people who are working in these disease areas or maybe related areas, like, but same organ, we could run screens there. Yeah, we'd be happy to hear from people. And yeah, Gordian.bio is the company website.
Amazing. Well, thank you. I really appreciate you taking the time. I learned a ton and really appreciate all the insight you've given to everybody.
Cool. Thank you.
That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Cynogenetics on Twitter, LinkedIn, or visit cynogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible. In particular, Amy Cousins and Sonia Shah, who produced the show, and James Pierce from Selective Frequencies, who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.